All right, folks, we're going to get started with our mitosis. It is going to be a review of what we've learned in grade 10. So it's nothing new. Hopefully this brings back a little bit of memory from what you've learned last year, or if you haven't learned it last year, maybe you'll learn something new today, all right? So we're going to take a look at mitosis and its stages. We're going to identify the stages of mitosis and explain the events that take place in each of the stages. Before we get into that, let's go back to what we looked at last time, where we looked at the cell cycle for eukaryotic cells that go through an orderly sequence known as a cell cycle that allows the cell to grow and replicate. Now, based on our the diagram that we looked at yesterday, it's very similar to the one that we have today. We can see that the cell cycle consists of two main stages. One is called interphase, that's the growing stage, and then we have the division stage, which consists of mitosis and cytokinesis. For more references on that, make sure you go back to the last lesson and take a look, okay? Because we go a little bit more in depth on that. So in our interphase, we get ready for mitosis during G1. Nucleolus can be seen, but now in S phase, or synthesis phase, our chromatin in the nucleus begin to condense and they turn into visible chromosomes, and your nucleolus disappears, and then you have your 23 pairs of duplicated chromosomes that start to move together. Okay, so here's a question. When do chromosomes duplicate? They duplicate during interphase before mitosis. And the purpose of that is so that the chromosome number of that specific organism can be kept the same. Recall from yesterday's lesson or 2 days ago, where we talked about how certain organisms have different number of chromosomes. Well, that's because that actually is the DNA that scripts for what that specific organism is. So if the chromosome number is wrong, then that means there can be issues with development for that organism. Okay, so let's get into the mitosis part here. Think back to last year. The 3 reasons are: 1, production of new cells; regenerate damaged tissues; and for maintenance. So every multicellular living organism start off as a single cell, but eventually become a makeup of billions of cells. And in order for an organism to grow, repair, and maintain its functions, it has to have new cells that gets created. So each cell that undergoes mitosis will produce 2 new cells, from 1 to millions to billions of cells that make up that organism. And throughout the process of mitosis and cell division, organisms can also regenerate damaged tissues. So think about the times when you injure your leg, or you sprain an ankle or something, or you get a cut on your arm. All of that happens immediately. Your cells will start to regenerate and your tissues will start to fix itself. That's why you see certain things like scar tissue, right? Because that's clear proof of when damaged tissues gets replaced and regenerated. Or another example is things like organisms like starfish. They can lose an arm due to a predator attack, and a new arm gets regenerated through the same process of mitosis and cell division. Now lastly, maintenance. So some cells need to be replaced because they can't function properly, or they have ended their life, right? So they have a timeline or how much they're going to live. So when they die, new ones got to replace them. So for us, we have cells like skin cells and blood cells get replaced regularly, and those cells go through mitosis and cell division as well. Now, when we talk about mitosis, it is strictly in our somatic cells. Somatic cells are body cells. These are cells that are not related to our sex. Sex cells are ones that have to go through fertilization, and the chromosomes are a little bit different there. And we're going to talk about meiosis, which is exactly what it is, in our next lesson. But for mitosis, it is exclusively for somatic cells. And in this situation, new cells that are produced are identical to the previously existing cells. So if they're specialized cells, they have to be the exact same type of specialized cells. However, if they're non-specialized or unspecialized cells, then they will remain unspecialized cells. We call those germ cells. And germ cells can go and be produced through meiosis into your sex cells as well. And we'll talk about that later on in this unit. Now, chromosomes within the nucleus hold the genetic information, and we learned that in the last 2 lessons. Each chromosome now is made of 2 sister chromatids held together by a centromere. And the sister chromatids are genetic copies of each other. If you can take a look at our chromosome here, it's a double-strand chromosome. It's attached by a centromere. And when they separate, you can see the sequence here. The pattern is exactly the same. And that's why we say these are sister chromatids, and they have exact same genetic copies of each other, because when they split up during mitosis, that's when they're going to form 2 new cells. So therefore, they have to be identical. Here's another example of how it looks like. Sometimes they can be illustrated as straight lines, but do know that there are different sequences and patterns. And we'll look at karyotyping, where we're going to see patterns and size and length is going to matter when we look at chromosomes later on. Okay, but do know that the centromere attaches them together, and the 2 sister chromatids will separate at some point. So the function of mitosis is to maintain the same number of chromosomes from cell to cell. And during the cell division, the original or parent cell divides into produce 2 new daughter cells. And each daughter cells have the same number of chromosomes as the parents, and then the daughter cells will eventually become the parent cells later on as they divide again. Now the 4 stages, PMET, prophase, metaphase, anaphase, telophase, these phases can be seen in rapidly growing areas of plants and animals. Okay, so stage 1, we have prophase. In this phase, chromatid condenses and thickens from visible duplicated chromosomes, and each chromosome looks like they're X-shaped. Each half of the X is 1 copy of the original chromosome, and since they are still connected, they're considered as 1 chromosome. At this point, you can start seeing the centrioles start to move to opposite ends of the cell. You can start seeing spindle fibers extend from the centrioles to the centromeres. The nuclear membrane starts to dissolve. This transitional period is called prometaphase, because it's transitioning from prophase to metaphase. Now during metaphase, meta means middle, so the spindle fibers attach to centromeres of replicated chromosomes, and they're guided. Chromatids are guided by spindle fibers to the middle of the cell. So we call that the cell's equator. So very similar to the globe or the Earth's equator, where it's right in the middle at the center of the cell. Now, a spindle fiber from one pole is attached to one chromatid, and the opposite spindle fiber or the opposite pole has a spindle fiber that attaches to the other centromere, so that when they shorten the length of the spindle fiber, later on they're able to separate from each other. Now what that does is each chromatid has now their own spindle fiber attachments to make sure that the division is going to be equal in terms of genetic information. What happens after this is anaphase, where the centromere now splits apart. Chromatids are pulled towards opposite poles of the cell by spindle fibers, and chromatids pull apart as spindle fibers shorten. And finally, we have chromatids arrive at the opposite poles of the cell. New membranes form around the daughter nuclei. Chromosomes will disperse and no longer are visible. Spindle fibers also disappear. And at this time, each chromatid is now a non-replicated chromosome. So they are separate now. Chromosomes are going to unwind and become less visible. And then before that phase ends, we get into cytokinesis. That's where our cytoplasm and all the other stuff that goes with it gets separated into 2 new daughter cells. Okay? Nucleolus reappears, and nuclear membrane forms around each new set of chromosomes. So everything gets split into half, a cell membrane forms, and after cytokinesis, 2 daughter cells are formed. So here are some images that you should have seen before. Let's look at some of these phases in real examples here. So early prophase, late prophase, as you can see here, you can see the chromosomes. And then now the chromosomes are even more obvious, and they're starting to take into their positions for metaphase. And then as they're in metaphase, they're all lined up at the center of the cell. So this one's very, very obvious here. And then as they pull apart, you can see two groups of chromosomes that are separated. So telophase here, the last one, we can see the two cells are clearly separating from each other. Here the cell plate is being produced into a cell wall later on to show that it is two separate cells. Okay, so that's it for our mitosis review. What you want to do is take a look at what we've learned in the last 3 lessons. Maybe go over it a little bit. We're going to get into meiosis, the formation of sex cells, in the next couple lessons. With that, we're going to have some sort of an assessment that's going to be coming up for us as well. Okay, so if you haven't been keeping up with our lessons, please take some time to maybe look over the slides again. And if you have any questions, make sure you find me, either email me or in the Q&A or our live stream. All right, take care, guys.